how are you doing? Welcome to PW English YouTube channel. I'm 𝐒𝐿𝐶𝐄𝐈𝐒𝐃, your botany teacher. I welcome you all to this lecture in the UMEED NEET English batch. In today's session, we are going to focus on the chapter Cell, the Unit of Life, which is a very easy chapter. Easy because it's not the first time you're learning something in this chapter. As the name itself indicates, in this chapter, we're going to learn about cells, its different types, structure, and all of that. Most of which you would have already learnt in your lower grades. So probably in your 8th, 9th and 10th grades. We will explore the same things in a little bit more detail in this chapter. So you will not be overwhelmed and shocked because you're learning something very difficult for the first time. And the questions that are asked from this chapter are also pretty easy and it'll not like mess up with your mind. You can score good marks and get all of the questions correctly answered from this chapter usually. Okay. So as always in this batch, we are going to discuss the whole chapter in one shot. We'll do that today for this chapter. And talking about the syllabus, here is a snip of what is the syllabus of this chapter for NEET 2025. Here is the snip of that NTA syllabus. Okay, so if you were to ask me whether you should refer old NCERT or new NCERT for this chapter, you could refer either. Unlike many other chapters where certain concepts are deleted, over here there has been no deletion absolutely. Whatever was there in your old NCERT is there in your new NCERT as well. So you could use either. Okay. We will learn about prokaryotes, eukaryotes, different kinds of organelles, structures, everything. Talking about its weightage. How many questions can be, expected in the NEET 2025 exam from this chapter? The range varies from 2 to 5 questions. There have been some years where only 2 questions have been asked. Also, there have been years where as many as 5 questions have been asked from this chapter. So you can expect anything between 2 and 5 in NEET 2025 also. And another thing, though we teach this under botany here, questions from this chapter can appear in your botany as well as zoology sections in your NEET exam. Okay. So without any further ado, let's get started. Let's talk about cells. Let's talk about everything about cells. So here's a first task for you. On the screen, there are a couple of images and I want you to pick whichever ones are living. There are living things and there are non-living things and you have to pick which ones are living. It's like a very easy task and you might be wondering why is she giving us some second grade activity. Yeah, it's easy, I know. Plant is living, this fish is living, this cute little puppy is living, pen, notebook, soil, sunlight and water, non-living. Now, what differentiates a living organism from a non-living thing? What is it that we have that non-living things don't have? What do I have that this pen does not have? What do I have that this watch strap does not have? What is it? The thing that all living organisms have, which non-living things may not have, are living cells, right? We are all made up of cells. So we have cells, non-living things will not have cells. For the same reason, we call cells as the fundamental functional unit of life, right? I'm sure it has been etched in your brains. You've been studying about the definition of cell probably since 5th, 6th grade. I'm sure your teachers also must have told you that it's an important question for board exam. Memorize the definition of cell. Cell is the structural and functional unit of life, right? We all know it. Why is it that we call the cell itself as the fundamental unit of life? Why do we call it as the structural and functional unit of life? If you take the levels in biological organization, you have many different types of biomolecules like proteins, lipids, and all of that. They make cell organelles. Many cell organelles are present inside a cell. Many cells come together and form tissues. Different types of tissues form organs and so on, right? Everybody knows about this. Why is it that we are calling the cell itself as the structural and functional unit of life? Why? Why not organelles? Why not tissues? Why is it the cell itself is the structural and functional unit of life? There are two reasons here. Firstly, a cell is the smallest unit that is capable of independent existence. Now, what do I mean by independent existence? You know, living organisms are of many different types. One way of classifying living organisms is based on how many cells are present in their body. Some organisms are made up of just one cell. Many organisms like us are made up of many cells. If they are made up of one cell, we call it unicellular organisms. If an organism is composed of more than one cell, then we call it multicellular organisms. Now think about unicellular organisms. Are they capable of independent existence? Do they perform all the activities that we do? Multicellular organisms, in us, there is digestion. We perform digestion, reproduction, excretion, growth, all of these. All of the same activities, metabolic reactions and all of these life processes basically, a unicellular organism also can perform, right? Take a unicellular organism, amoeba for example. Amoeba, does it undergo digestion or how do you say it? Does it perform digestion? Yes, it will engulf food particles, it will break it down. Digestion happens even in unicellular organisms. Does it perform reproduction? Yes, even they undergo cell division, they also multiply, increase in number. Do you see excretion in unicellular organisms? Yes, they may not have specialized systems because they're just made up of one cell, but you do see there is excretion of waste material even in amoeba. So all of the life processes that we can perform, a single cell also can perform, and it can exist independently. Can organelles exist independently? Just a mitochondrion, just Golgi apparatus, or just endoplasmic reticulum? Can it exist independently? It cannot. The smallest unit that can exist independently in biological organization is a cell. Example for that is our unicellular organisms. Of course, tissues, in some organisms you see tissue grade organization or tissue level of organization. But over there, it's not the smallest unit capable of independent existence. Of course, it also can perform all of the cellular activities and basically life processes, but it's not the smallest unit. The smallest unit in biological organization that is capable of independent existence and performing all life processes is a single cell, which is why we call cell as the fundamental, structural and functional unit of life. All right? Yeah. Like I've already mentioned, anything less than a complete structure of a cell, if it is anything less than a complete cell, just the organelles or just some parts, not the complete cell, it cannot exist independently. All right? That's the reason why we call cell as the fundamental, structural and functional unit of life. How do we know about cells? Who are the first people who researched about cells? And that is something that you should know about the history of discovery of cells. Two people's names you'll have to remember here. One is Anton van Leeuwenhoek and the other one is Robert Brown. This person, Anton van Leeuwenhoek, is the first person to observe living cells under the microscope. Before this, Robert Hooke did observe cells, but they were cells of the bark like the cork. They were dead cells. But he is the person to have first observed living cells under the microscope. The cells that he observed were tiny animal cells like protozoans, microscopic animal cells. And he calls them animalcules because they're like tiny animals. And the first person to have discovered the nucleus is Robert Brown. Okay, about these two scientists' names and what is your contribution, you should remember. There's something called cell theory we must learn about. Okay, this theory was formulated by three people. We learn about their contributions. First, Matthias Schleiden. Matthias Schleiden was a botanist from Germany. Like all botanists, he also studied plants. But he studied plant cells by looking at them under the microscope. And he saw that the tissues or the cells in plants there were many different types of cells. He noticed that within plants you have many different types of cells, and those different types of cells formed tissues of the plant. This was his observation. We've already learnt about it in our previous chapter, Anatomy of Flowering Plants. Within plants, you do have different types of cells that are forming different kinds of tissues. So that was the observation of Matthias Schleiden, a German botanist, in the year 1838. Around the same time, in the year 1839, another researcher, a zoologist, okay, his name was Theodor Schwann. He was a zoologist, of course. He was studying about animals, and he was also interested in looking at plant tissues and animal tissues under the microscope. What he says is, of course, he's a zoologist, he's studying animals, but he also looks at plant tissues under the microscope, and he notices like a very clear, distinct difference between plant cells and animal cells. He sees that in plant cells and animal cells, in both of them, there is something like a cell membrane. But a plant cell, apart from the cell membrane, contains an other layer outside, which is called the cell wall. He notices how that cell wall is present only in plants and not present in animals. Okay, so he distinguishes plant cells and animal cells by the presence of cell wall in plant cells. Okay, and with his knowledge combined with what Matthias Schleiden also had observed and published, he comes up and formulates what is called the cell theory. So in the cell theory, he states that all living organisms, be it plants or animals, okay, in all living organisms, plants and animals, they are composed of cells and its products. So he says, all bodies of plants and animals are composed of cells and products of cells. Cells, okay, you understand. But what are these products of cells? This can have two connotations. Firstly, product of cell means cells coming together to form tissues. Product of many cells. That could be one thing, one way of thinking about it. Another thing is, of course, we will have cells. And along with that, we have products of cells, which is something that the cells will produce. Like it could be some secretions of cells, right? So that's another way to think about it. So what he says is, all living organisms, plants and animals, our bodies are composed of cells and its products, which is right? And then there was one other question. All right, I get it. All living organisms, plants and animals, we're composed of cells and its products. But when an organism grows, the number of cells in the body will increase, right? How many cells we had as a little baby? We don't have the same number of cells as adults. So when a little baby grows to become an adult, as it grows in size, the number of cells is going to increase. A small sapling growing into a tall tree, the number of cells is obviously going to increase. When new cells are added to a plant, where is it exactly coming from? How are new cells formed? This was one question which was unanswered by their hypothesis of the cell theory. So there, another person called Rudolf Virchow, how are new cells formed? Rudolf Virchow explains that in the year 1855. He says that in an organism, in a body, if a new cell has to arise, then there's only one way it can happen. Only if already existing cells undergo cell division, only then newer cells can arise. Just like that, by magic, new cells cannot be made. So newer cells can only arise from pre-existing cells by cell division. So initially probably a couple thousand cells over there, they will undergo cell division, give rise to more cells, and that is how growth will take place. He says that cells arise from pre-existing cells. They include this statement also in the cell theory, and in Latin it is omnis cellula acellula, which means cells arise only from pre-existing cells. New cells can arise only from pre-existing cells. So whatever hypothesis was formulated by Schleiden and Schwann, which said animals and plants both contain cells and their products, he modified that a little bit, and along with that statement, he added this statement as well. And today we know cell theory as these two statements. All organisms are composed of cells and its products. New cells arise from pre-existing cells. Okay? All right. That's about the cell theory. Now, let's understand different types of cells. Cells are of two basic types based on whether the DNA or the genetic material is bound by a nuclear membrane or not. Okay. Now, in some type of cells, of course, you know that in all living organisms, bacteria, eukaryotes, in every one of us, DNA is the genetic material. Viruses are the exception. In few viruses, RNA can be the genetic material, but in prokaryotes and eukaryotes, DNA is the genetic material. If you consider our cells and plant cells, the DNA, because it's so precious, it has so much important information, it's very well protected inside a membrane-bound nucleus. Whereas in some cells like bacteria, the DNA is not covered by a nuclear membrane. So based on this one basic difference, you can classify living organisms into two types. If they are organisms that are whose DNA is not bound by a nuclear membrane, then we call it prokaryotic organisms. Over here, pro means primitive, ancient, like olden days, like really ancient type of nucleus. Karyotic, karyon means nucleus, like a really primitive nucleus. What is the characteristic feature of these organisms? The nucleus, there is DNA, but around this DNA, there is no membrane. Okay? What about eukaryotic cells? U means true. Karyon again means nucleus. The difference here is, here you have the genetic material inside. This genetic material is surrounded by a well-defined nucleus. Let me use a different color. A well-defined nuclear membrane. This is the primary most important difference. They have a membrane-bound nucleus. What is common to them both? In both prokaryotes and eukaryotes, within the cell, there will be a semi-fluid matrix called the cytoplasm. Cytoplasm is common to both. Okay? And then this cytoplasm is what will occupy the most volume inside the cell. That is where most of the cellular activities, metabolic reactions will take place. So that is common to both these organisms or both these types of cells. Okay, now another difference, the second difference. This was the first primary difference. The second difference is prokaryotes don't have a membrane-bound nucleus and they don't have membrane-bound organelles also. Membrane-bound organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, lysosomes, none of these membrane-bound organelles are present in prokaryotes. You can see membrane-bound organelles being present in eukaryotes. Okay, so they lack the membrane-bound organelles. But here they do have membrane-bound organelles like endoplasmic reticulum, Golgi, mitochondria, lysosomes. All of these are membrane-bound organelles. Okay, so you can see how this is Golgi apparatus. It is bound by a membrane. What I have highlighted here right now is membrane. Even in ER you have membrane. Even in mitochondria you have two membranes in fact. So all of these lysosomes have membrane. All of these are membrane-bound organelles. You do not find membrane-bound organelles in prokaryotes. That's the second difference. Does that mean there is no organelle that is common to prokaryotes and eukaryotes? There are no membrane-bound organelles here. There are membrane-bound organelles present here. There is one organelle that is common to both prokaryotes and eukaryotes. And that is the ribosome. I told you that prokaryotes lack membrane-bound organelles. Now I am saying that prokaryotes have ribosomes. What does that mean? That means ribosomes don't have a membrane. Ribosomes are not membrane-bound, right? So ribosomes are not membrane-bound organelles. They are found in both prokaryotes and eukaryotes. Though it is common to both of them, still there is some difference in the structure between prokaryotic and eukaryotic ribosomes. When we study about the structure of ribosome and its function, we'll understand that. But because ribosomes are a part of prokaryotic cells as well as eukaryotic cells, it's found in both prokaryotic as well as eukaryotic organisms, we call ribosomes universal organelle. It's present in all types of organisms, be it prokaryotic or eukaryotic. Okay. So first difference was there is no membrane bound nucleus, there is membrane bound nucleus, no membrane bound organelle, membrane bound organelle is present, organelles, many of them are present. Here ribosomes are there, here also ribosomes are there, but still there is difference in the structure of ribosomes. We'll learn about it later. Okay. Now let's talk about the different shapes and sizes of cells. Generally, prokaryotic cells are smaller in size compared to eukaryotic cells. Here, let's talk about the size of certain cells. What you're looking at here is mycoplasma. You've learnt about it in biological classification under Monera. I told you there is a wall-less prokaryotic organism called mycoplasma. It can cause diseases and all of that, right? So this is that mycoplasma. Its length is about 0.3 microns. Here you see a bacteria, which is a rod-shaped bacterium. Its length varies between 3 to 5 microns, which is like super tiny. And then human RBCs, the diameter is around 7 microns. Okay, so the size is variable. Neurons are some of the longest cells. In fact, in our body also, neuron is the longest cell. Apparently, there is a neuron that runs from the tip of your spinal cord, from the base of the spinal cord near your like tailbone till your big foot, like the thumb in your leg, like the big toe or whatever you call it, big toe I think. Yeah, from the back, from your tailbone, from around that area, it runs down your leg till the tip of your big toe. So that's how long just one cell can be. Of course, there are variations. Every cell in our body is not the same length. Every cell in our body is not the same shape. Basically, what is the size of a cell? What is its shape? Depends on what its function is in our body. Let's understand that. About size, I've already told you. Let's talk about the shape now. Many different types of cells come in many different types of shapes. We've learnt about the different types of cells and its shapes in plants in the previous chapter, anatomy of flowering plants. Even in animals, if you think about our body, if you've already learnt structural organization in animals, tissues if you've learnt, you will already know that even in our body, we have so many different types of cells. We have squamous epithelium, you have cylindrical, skeletal muscle cells, and then you have columnar cells, and you have so many different types of cells, cuboidal epithelium. It comes in so many different types, and RBCs are biconcave. Neurons have this crazy head with like one crazy hair and one long tail. So they come in so many different shapes. The reason they are in so many different shapes, why they are in a particular shape, is because that shape helps them to perform whatever function they're supposed to do. For example, if you consider RBCs, RBCs are biconcave, right? They are like this. They're round and biconcave. Now, why should RCBs be biconcave? Why can't they be just spherical and round and big? That's because if they are any shape other than this, they may not be able to do their job well. What is the job of RBCs? RBCs are present in our blood. Our blood transports respiratory gases across different parts of the body, oxygen, carbon dioxide, and also transports nutrients, right? In blood, RBC's job is to help in the transport of respiratory gases like carbon dioxide and oxygen. Within RBCs, there are molecules called hemoglobin, which will help in the transfer of oxygen. Okay, so if this RBC has to efficiently transport oxygen to the tissues, I want you to think about it. You have different sized blood vessels. Whatever oxygen you are inhaling right now is going into your lungs, going into your alveoli, and then is going to get transported into your capillaries. Tiny, tiny blood vessels carry that oxygen via hemoglobin present inside RBCs majorly and take it to different parts of your body, delivering oxygen to every one of your tissues. So if RBC has to perform that job very efficiently, it requires a lot of space to accommodate hemoglobin molecules inside. If there's one big fat nucleus sitting in the center of the cell, RBCs will not have sufficient space to accommodate RBCs to, help in the transport of respiratory gases. So as RBC matures, it will lose its nucleus. Once the nucleus is gone, once the cell becomes enucleated, what was initially spherical will now become biconcave. So now there is additional space available for accommodating more hemoglobin molecules. Also, this shape allows RBCs to squeeze in through the tiny capillaries and deliver respiratory gases or pick up respiratory gases from there. Okay, so it's because it has to function in that way, it has modified its shape accordingly. Okay, and then white blood cells, why are they amoeboid? They are amoeboid because what is the function of white blood cells? They are involved in protecting us. They are part of our immune system. Whenever there is a cut or a bruise or an infection, they are the cells that will first go to that side and try and fight that infection or the pathogen, right? So in order to do that, let's say there's a cut in my skin, bacteria have entered my body, then these WBC cells will have to come from within the blood vessel outside to the site where infection has happened. For that, they will have to squeeze in through the capillaries. If this is the capillary wall, which is lined by squamous epithelium, these white blood cells will have to squeeze out of this and go to the site of infection. Okay, for that they should be able to change their shape and be amoeboid. If they are very rigid, then they will not be able to do their job well. Likewise, columnar cells or nerves being like so branched on top and having a long tail because it has to transfer information really quickly. Long tracheid or trachea for that matter, and it's empty inside because that helps in xylem transport. If it was a living cell and it was very short, there would be a lot of resistance for transport of water happening through xylem against gravity along long distances. To ease that whole thing out, they have this shape. And mesophyll cells are round and oval, have a lot of chloroplasts because they have to perform photosynthesis. So a shape of any cell is the way it is, not because it likes that shape, because it helps that particular cell function more efficiently because of that shape. All right, now let's talk about prokaryotic cells. So what do we already know about prokaryotic cells? They have a primitive nucleus. They do not have a nuclear membrane surrounding the genetic material. They lack membrane-bound organelles. All they have is just one organelle called the ribosome. Which are the organisms we will include under prokaryotes? Can you remember in the chapter biological classification, we have studied about a kingdom Monera, which includes exclusively prokaryotes. Over there, we've learnt about bacteria, archaebacteria, eubacteria, cyanobacteria, and all of those different types, mycoplasmas. So all prokaryotic organisms are monerans. So that includes mycoplasma, bacteria, and blue-green algae. Blue-green algae is also called cyanobacteria. Okay? Yeah. So how are these prokaryotic cells different from eukaryotic cells when it comes to size? I've already told you that in comparison with eukaryotic cells like RBCs and WBCs, the prokaryotes are extremely small. Here it is given that E. coli, a type of bacterium, measures in length just about 2 microns. Microns are like really small. 1 millimeter, I'm sure you know 1 millimeter in your ruler that you use. 1 centimeter will have 10 millimeters. 1 millimeter, in 1 millimeter, if you make like 1000 equal divisions, then one small division will be 1 micron. So 2 microns is like super tiny. So bacteria are small and relatively our eukaryotic cells are larger. When you talk about how frequently they undergo cell division, prokaryotes divide more rapidly, they undergo cell division more frequently than the eukaryotic cells. Okay? Yeah. Now, talking about their shape, prokaryotes vary in shape. They come in so many different shapes and sizes. Here is a diagram from NCERT textbook. Okay, bacteria 1 to 2 microns, PPLO is 0.1 microns. Alright, in comparison with eukaryotic cells, they are like super tiny. Bacteria come in 4 shapes. About this we've already discussed in the chapter biological classification also under bacteria. These are the 4 major shapes. If it is spherical, you call it cocci. If it is rod shaped, it's bacillus. If it is spiral, it's spirillum. If it's comma shaped, it's vibrio. Okay, so these are the 4 basic shapes in which you can see bacteria. Coccus is singular, cocci is plural. Bacillus is singular, bacilli is plural. Vibrium is singular, vibrio is plural. And spirillum is singular, spirilla is plural. Okay. Talking about the structure of bacterial cells. Whenever we study about the structure of anything, we always start from the outside and move towards the inside. In a bacterial cell envelope, you have 3 distinct layers, okay? So, the covering, outer covering of the bacterial cell is called the cell envelope. It has 3 distinct layers and it has certain modifications as well, certain appendages that arise as well. So, we will talk about it, okay? So, first, they have a complex cell envelope. It is a 3-layered structure. I told you that. Though there are 3 distinct layers, ultimately the function of any cell envelope is to offer protection. So, though there are 3 different layers, together they act as a protective unit. They offer protection for the cell. That's what all walls do, right? The wall of your house, the wall of your room, they basically offer protection. Even in cell walls of prokaryotic cells, there might be 3 distinct layers, but ultimately they all offer protection for the bacteria. Which are these 3 cell layers? Let's understand from outside to inside. The outermost layer is called the glycocalyx. Right inside the glycocalyx, you have the cell wall. Inside the cell wall, you have the cell membrane. Okay, so outermost is the glycocalyx. In the middle, you have the cell wall. Right within the cell wall, the innermost layer is your cell membrane or your plasma membrane. Let's understand what these different structures are made up of. The glycocalyx is made up of polysaccharides. In different species of bacteria, which polysaccharide it is made up of can vary. Okay, it's not like in all bacteria, glycocalyx is made up of the same substance. It's of course made up of polysaccharides, but which polysaccharide it is made up of can vary among different groups of bacteria. Also, what varies is its thickness. Its composition can vary. Composition means what it is made up of, which type of polysaccharide it's made up of. Along with that, how thick it is also varies between different bacterial groups. In some of them, it's really thick and it's very tightly attached to the cell wall. In such cases, it becomes difficult to remove the glycocalyx layer, wash off the glycocalyx layer. While in some other cases, the glycocalyx will be a very thin layer. In such cases, it can be removed easily. It is not as tightly attached to the cell wall as the thick layer. Whenever it is thick and very tightly bound to the cell wall, we call it capsule. Okay? It's called capsule when it is thick and tough. Whenever it is loose and not so tightly bound, you call it slime sheath. I repeat, outermost layer. Here you can see there are 3 distinct layers. I will show you. This outermost one is in a sort of bluish color. That is your glycocalyx. Right inside that, you have the cell wall. Inside the cell wall, you have the plasma membrane. Right? Glycocalyx outside, cell wall middle, cell membrane inside or plasma membrane inside. What is the composition of glycocalyx? Generally, it's made up of polysaccharide. But which polysaccharide exactly? You cannot generalize it because in different groups, it is different. Is this glycocalyx the same thickness in all bacteria? Not really. In some of them, it's really thick and tough, tightly bound. In some of them, it's thin and loosely bound. If it is thick and tightly bound, what are we going to call it? We'll call it the capsule. If they are thin and loosely bound, then we call it the slime layer. Okay, or you can call it the slime sheath. That's about your glycocalyx. We all know that monerans have cell walls, right? We learnt about it in biological classification. Bacteria generally in most cases do possess cell walls that is composed of a substance called peptidoglycan. It is made up of peptidoglycan. But if you remember when we discussed Monera, at the end we spoke about mycoplasma in the chapter biological classification. I mentioned how Despite being prokaryotic like bacteria, mycoplasmas lack a cell wall. They do not have a cell wall. Then why are we calling it prokaryotic? Because it does not have a membrane-bound nucleus. So except for mycoplasma, every other prokaryote will have a cell wall, and that cell wall is composed of peptidoglycan. What is the function of the cell wall? What is the function of cell wall generally? It will give a proper shape for the cell, it will offer protection as well, and it will provide structural or mechanical support. So it determines the shape of the cell and it also provides a strong structural support. Why is that necessary? It's necessary so that the bacterial cell, if it's aquatic or anything, does not take up too much water, burst and or collapse. So such things should not happen. Therefore, the cell wall acts like a protective layer. Now, let's talk about the innermost layer in the cell envelope, which is the cell membrane. The cell membrane here is very similar in structure to how the cell membrane is in eukaryote. So, over here we will not discuss the details of its structure. In eukaryotic cells, cell membrane, when we are discussing, I will explain to you what are the different chemicals or molecules that you can find in the cell membrane. Exact same structure will exist even in prokaryotes. So that is common. Cell membrane is common between prokaryotes and eukaryotes. And this cell membrane is a selectively permeable membrane. What does that mean? It is permeable, of course, which means it allows certain molecules to transfer across it. If there is certain molecule that have to move from outside to inside the cell or from inside to outside the cell, it will allow movement of it, but not everything and anything. It will selectively allow certain molecules to move into and out of the cell. That's why we say it is selectively permeable. Okay? And yeah, structurally, like I already mentioned, it's very similar to eukaryotic cells. So the cell envelope outermost has glycocalyx made up of different types of polysaccharides. Middle you have cell wall made up of peptidoglycan. Innermost you have the cell membrane. And the cell membrane structure we're not discussing right now. We learn about it in eukaryotes. It's pretty much the same thing. It's exactly the same thing. Okay? Yeah. Now depending upon how the cell wall is, the structure of the cell envelope basically, not necessarily the cell wall, the cell envelope basically, and how a bacterial cell responds to a type of staining technique called gram staining. You know how if you have to look at cells under the microscope, you have to add color to it and then you can visualize the cells very well under the microscope. So there are different types of staining procedures that you can do before you look at a cell under the microscope. One such staining procedure was developed by a person called Christian Gram. That's why it's called gram staining. This is not kilogram, milligram kind of gram. This is the name of a person. So you don't have to learn about how exactly that gram staining has to be done. In the gram staining procedure, there is a series of steps that you need to perform. There is a stain used, crystal violet it's called, it's also called the gram stain. It is blue in color. When you add this stain in the series of steps, in one of the steps if you add it to bacteria, at the end of gram staining, some of these would have retained the gram stain and they will appear blue in color. Some of them would have lost the blue color gram stain and they appear somewhat reddish in color. Okay. The reason is because of the difference in structure of the cell envelope. If you see gram positive bacteria, the reason why we call them gram positive bacteria is because even after all the steps are completed, it still retains the gram stain, which is blue in color. Why we call bacteria gram negative is because you would have added a gram stain, violet color at the beginning some point, but after a while, it will lose that color. It will become some other color. Okay, it will not retain the blue color basically. At the end of the procedure, it will not retain the blue color. That's why there is no gram stain retained. That's why you call it gram negative. Okay, so don't worry about the details of how it is performed. It's not part of your syllabus. So the difference, why one retains the gram stain, why one doesn't retain the gram stain and is called gram negative is because of the structure of cell envelope. In both of them, you will have a cell wall made up of peptidoglycan, you will have a plasma membrane. Okay, but in gram positive, I want you to notice how whatever is shown in purple or blue color here represents the cell wall peptidoglycan. I want you to notice how in gram positive the peptidoglycan layer is thick, while in gram negative the peptidoglycan layer is thin in comparison. Okay, this is one difference. And look at the cell membrane. It is common in both. There is no difference here. In gram-negative bacteria, in addition to the plasma membrane inside the peptidoglycan layer, you will have a plasma membrane-like structure outside the peptidoglycan layer as well, and that is called the outer membrane. This outer membrane you do not have in gram-positive bacteria. Okay, so that's the difference. In gram-positive, peptidoglycan is thick, gram-negative peptidoglycan is thin. Gram-positive doesn't have an additional outer membrane, while gram-negative does have an additional outer membrane. Okay, so based on these structural differences, because of these structural differences, whenever gram staining is done, this will retain the gram stain, and that's why we call it gram-positive. This will not retain it, that's why we call it gram-negative. Okay, now let's talk about some membranous structures that we find in bacteria. Okay, I told you that in bacteria or prokaryotic organisms, you do not have a membrane-bound nucleus, you do not have membrane-bound organelles. If you do not have membrane-bound organelles like mitochondria, which are responsible for generating energy in large amounts, mitochondria is the powerhouse of the cell. Everybody knows mitochondria is responsible for the formation of ATP majorly inside the cell. Everybody knows about it. ATP is the energy currency of the cell. Everybody knows about it. If the cell has to synthesize additional copy of DNA, if the cell has to make proteins, if the cell has to synthesize RNA, anything, if any synthesis reaction has to happen, anabolic reaction has to happen inside a cell, it requires energy. And this energy in eukaryotic cells is majorly provided by the mitochondria. But in these prokaryotes, mitochondria itself is not there. If mitochondria itself is not there, then how is ATP produced? Where can ATP get produced? Because even in prokaryotic cells, protein synthesis has to happen, RNA synthesis has to happen, DNA replication has to happen. All of these are energy consuming activities. If there is no mitochondria, then where do ATP synthesis, where does it take place? It takes place in a structure called the mesosome, right? Never mind. So what basically happens here is, I'll show you. If you can think of this as our mitochondria in eukaryotes, you know how the inner membrane of mitochondria has many infoldings called cristae, right? So it is within these cristae that there are enzymes that synthesize ATP. Now, if you consider like a bacterial cell has an outer cell wall, and inside the cell wall, it has a cell membrane, right? The structure of this membrane here and the structure of this membrane is both the same. The structure is the same. So instead of these bacteria having a specialized extra organelle to synthesize ATP for it, within the membrane that it has, the only type of membrane that it has, which is its plasma membrane, it will make some modifications. It will develop certain infoldings here within the plasma membrane itself, which we refer to as the mesosome. And in these infoldings or invaginations, the enzymes required for ATP synthesis will be present. So the plasma membrane itself will get modified in certain regions and form the mesosome. Within that, you will have ATP synthesis, enzymes required for ATP synthesis. You might be wondering, why should it cause infoldings there? Why can't ATP synthesis, the molecule required for ATP synthesis, be present right here? The reason is because if it is enfolded like that, it increases the surface area of plasma membrane. It can accommodate more ATP synthase molecules so that more energy can be generated. That's the same reason why our mitochondrial inner membrane is also having so many infoldings. Because of those infoldings, you can see how many such enzymes can be accommodated. Though there is no special membrane bound organelles, with whatever membrane it has, bacteria does a beautiful job of modification according to its needs. Okay, yeah. So these are basically extensions of the plasma membrane. into the cell, into the cytoplasm. So here is an illustration. What you're looking at here outside is the cell wall. Right inside the cell wall, you will have the cell membrane. The cell membrane has unfolded like this, right? That is forming the mesosome. Within the mesosome, you can see structures like vesicles, tubules and lamellae. Okay? Vesicles are tiny structures like this. These are the tubules and you will have lamellae. Basically, they are the membranes. Okay? So that's about the mesosome. Mesosome doesn't help only in cellular respiration or synthesis of ATP. That of course is an important role of mesosome, but apart from that it helps in a lot of other things like helping the DNA replicate before cell division. After DNA has replicated, the cell has to divide. It will help in the formation of the cell wall. Secretion process also it helps. If something has to be secreted, because of these enfoldings, the surface area available will be more. Alright, so all of these are some functions of the mesosomes. Talking about another membranous structure called the chromatophores, these are found specifically in bacteria that perform photosynthesis. Which are those bacteria that perform photosynthesis? It is the cyanobacteria, right? So the cyanobacteria are photosynthetic organisms which are prokaryotic. They do not have a membrane-bound nucleus. They do not have membrane-bound organelles. So they don't have chloroplast because chloroplast is a membrane-bound organelle. If they don't have chloroplast, then how can they perform photosynthesis? Why do green plants, higher plants require chloroplast? Because it is within chloroplast that the pigment required for photosynthesis is present. Chlorophyll is located within the chloroplast. That's why we need a chloroplast. But here, instead, what happens is, if this is like the bacterial cell wall, right within the cell wall, you will have the cell membrane. The cell membrane itself will form certain enfoldings like this, which are called chromatophores. There will be a mesosome here, let's say. And within these enfoldings, you will have pigments required for photosynthesis. So these invaginations where you have pigments required for photosynthesis, these are called chromatophores. Eukaryotes have a specific organelle to perform photosynthesis, the chloroplast. But here, since the membrane-bound organelle chloroplast is not there, the only membrane they have, which is the plasma membrane, itself modifies itself in certain locations. And in those areas, those regions of the plasma membrane, you can find the presence of pigments required for photosynthesis. Those structures can be called chromatophores. Okay, so these are found in not all bacteria, only in photosynthetic bacteria, that is cyanobacteria. They are also membranous extensions into the cytoplasm and they contain the pigments required for photosynthesis. Is that clear? Cool. Let's move on. Now, let's learn about flagella. Flagella are locomotory structures. Not all bacteria are motile. Some bacteria have the ability to move. Some bacteria don't have the ability to move. Whichever bacteria is motile or has the ability to move, they will have flagella as the locomotory structures. And it's not necessary that in all flagellated bacteria, you should have a fixed number. Some of these bacteria have only one flagellum. Some of them have two on either side. Some of them have like a tuft of them on either sides. Some of them, throughout the surface, you will have flagella. So it differs from bacteria to bacteria. But overall, whichever bacterium is motile, in that you will 100% find flagella as the locomotory structure. So its number and distribution in a bacterial cell can vary. This flagella is made up of a protein called flagellin. Okay, it's made up of even in eukaryotic cells you can find flagella, but the structure is very different. We'll learn about that also when we discuss eukaryotic flagella, okay, eukaryotic structures. But for now, understand that the structure of flagella in bacteria is super simple. You have just three parts. A basal body right here. I will highlight in pink or maybe blue. So you can see this thing here. This is called the basal body. The basal body is present in the plasma membrane. From the basal body, the flagellum will arise. After the basal body, you have a region called the hook, which is kind of flexible. It will twist around and turn around and help in the movement of the flagellum. So this is the basal body. Here you have a hook, and the longest portion of the bacterial flagellum is called the filament. Basal body, hook, filament, three parts of bacterial flagella. It's composed of flagellin. Its number and arrangement can vary in different bacteria. Okay, so that's about the flagellum, which is a locomotory structure. Next, let's learn about pili. Singular is called pilus. Okay, so these are also structures that arise from the surface of bacteria. This is made up of a protein called pilin. Okay, basically its function is not mentioned in NCERT, but they are involved in a type of reproduction, sexual reproduction, very primitive type of sexual reproduction called conjugation. So generally bacteria will have pili like this. So one strain of bacterium will have a pilus, one strain of bacterium will not have it. This will have it, let's assume. So this pilus will attach with the one that does not have the pilus. It will form like a channel through which its genetic material can be transferred to this bacterium. So in bacteria, you know that exchange of genetic material itself is considered equivalent to sexual reproduction. You do not have specialized gametes or anything formed. So here this is considered to be sexual reproduction, and for conjugation pilus is used. Okay. Next we have something called fimbriae. Fimbriae are very small multiple bristle-like structures that sprout from the surface of bacterial cells. You know, in bacteria, when they grow and form colonies. So in colonies, each bacterium is attached to another. For attachment, for adhering to the other bacteria, these fimbriae play an important role. And if the bacterium is a parasite, it has to attach to the body of the host. Then with the help of fimbriae, it can attach. It helps in attachment. Or if it's growing on rocks or something that are slimy, it will help in attachment of bacteria. Okay, so the fimbriae are very small bristle-like structures. You know what bristles are like, right? So it's like the bristles of your toothbrush. They are short and you will have many such bristle-like structures sprouting out. They are relatively shorter compared to your pilus, but more numerous. They are more in number. So you have, see, this in pink is the pilus. The shorter bristle-like things that I am showing in blue here, these are the fimbriae. They are bristle-like fibers. They help attach bacteria to the rocks in streams and also to the host tissues, like I already mentioned. Alright, now let's talk about ribosomes in prokaryotes. Ribosomes are structures within which proteins are formed. They are found both in prokaryotes as well as eukaryotes. The ribosomes are, they have two subunits. One is called the large subunit and the small subunit. Here also, even in prokaryotes and eukaryotes, ribosomes will be composed of two subunits, the large and the small. Here in prokaryotes, the large subunit is 50S. The small subunit is 30S. Unless they are synthesizing proteins, which is their job, they do not stay together. They can exist as separate units. But whenever protein synthesis has to happen, then the two subunits will come together. When they both come together, it is called 70S. It becomes 70S when they are together. I want you to notice how 50 plus 30 is not 80, 50 plus 30 is 70. Because here it is not additive. First of all, let's try to understand how did we come up with these numbers. Why is it 50 specifically? Why is it 70 specifically? How do we come up with these numbers? So, Basically, the S here represents sedimentation coefficient. What we do here is you take the ribosomes, you put it inside a centrifuge tube, you keep the centrifuge tube inside the centrifuge instrument and you spin it at very high speeds. You know, in a centrifuge instrument, basically centrifugation is happening at very high speeds. There is spinning, like probably 50 to 75, sometimes 100,000 times it will rotate per minute. Like they will rotate it like that for a specified duration of time. Then according to their densities, according to their size, they will start separating out. Okay, whichever ones are more dense, they will settle at the bottom of the tube. Whichever ones are less dense, they will settle somewhere in between. So depending upon how quickly they settle, you give them units. The ones that are heavier will have a higher number. Okay, so let's say this is the centrifugation tube. Inside this you've added ribosomes, small subunit, large subunit, and together everything you've added. Because this is the most dense, this will settle here. Because this is relatively less dense, it will settle here. This is even less dense, it will settle here. So whichever is the heaviest, most dense, that will settle at the bottom. That is what happens when you mix like some sand grains in a glass of water and spin it, right? Because of the centrifugal force, what will happen is whichever is more dense will settle at the bottom. So the larger soil particles settle at the bottom, the smaller ones will still be suspended, floating here and there, and eventually that will also settle down. But what I'm trying to say here is whichever ones is more dense, that will settle down quicker. Whichever ones have less density, that will take slightly longer to settle, right? So based on that, we give it units. Similar to how you have in your ruler, this much is 5 centimeters, this much is 10 centimeters. Even here, we will have at one, if it gets like deposited here, if it settles down here, this would be like, let's say 70s. If it gets deposited here, this will be 60s. Here it is 50s. Here it is 30s and so on. So depending upon where it will settle down in this tube, you give it these units. The reason why these are not additive is something that physics can explain to you. You know how when two objects are brought together, combined together, their overall density will reduce. So that is what happens even over here. That's why we will get less number. So 50 plus 30 here is not 80, it is 70s. Okay, so there are two subunits. The large subunit is 50s, the small subunit is 30s, and together it is 70s. All right. Sometimes what happens is during protein synthesis, when proteins have to synthesize, get synthesized, they get synthesized inside the ribosome only. Okay, that's why we call ribosomes as the protein factories. So when ribosomes have to synthesize proteins, this is something you will learn in upcoming chapters in molecular basis of inheritance, how exactly it happens. For now, just understand. There is a molecule, a type of RNA called mRNA. This mRNA will have the message for protein synthesis. What kind of protein has to be synthesized? That message mRNA will get from the DNA. Okay, so these ribosomes will start reading the message on mRNA and moving from one side to the other. From one end to the other, it will start moving. It is possible that sometimes one single mRNA is being read by so many ribosomes, and all of these ribosomes are simultaneously synthesizing proteins. Such a structure is called a polysome or polyribosome. Poly means many. Many ribosomes attached to one same strand of mRNA, that is called a polysome. Okay? Alright. Last part of prokaryotic cells. They don't have any membrane bound organelles, but they have certain structures called inclusion bodies within the cytoplasm, which are basically granular in nature, and they help in storing reserve food. Some examples you'll have to remember for that. You have phosphate granules, synophysin granules, and glycogen granules. Depending upon how food is stored in different types of bacteria, they can have these granules. Okay, so they are not bound by any membrane. I've already told you multiple times that in bacteria, in prokaryotes, there are no membrane bound structures, absolutely. In aquatic fungi, which are generally photosynthetic like your cyanobacteria, or even green sulfur bacteria and purple sulfur bacteria, if they are photosynthetic, they are generally aquatic, they will grow in water. In order to help these bacteria float on the surface of water so that they can get sufficient amount of sunlight for photosynthesis, there will be gas vacuoles present in them. Even they are not bound by any membrane. Basically, they help in, they provide buoyancy to the cells so they can float on the surface. That's all about prokaryotic cells. Now let's move on and learn about eukaryotic cells. Everybody is ready? I hope you said yes. So which are the organisms that we include under eukaryotic cells? We've already discussed this. In the chapter biological classification, we have protists, plants, fungi, animals. All of these groups, they are eukaryotic. They have a very well defined nuclear membrane. A membrane bound nucleus is present. They have membrane bound organelles as well. So the structure of eukaryotic cells is relatively much more complex than a prokaryotic cell. Let's explore it. All right. So it includes protists, fungi, plants as well as animals. In eukaryotes, you already know the nucleus is well defined. The nucleus is covered by a nuclear membrane. They do possess membrane bound organelles. I've already told you that. Eukaryotes, just because they are eukaryotic doesn't mean all eukaryotic cells will have the same kind of structures and that they are identical. In your lower grades, you may have already studied the difference between a plant cell and an animal cell. Even in this class, I've told you in the beginning, right? So here is an illustration of a plant cell. And here you have an animal cell. Okay. So plant cells have a cell wall, which animal cells don't have. Plant cells have a large central vacuole, which animals don't have. Plant cells have chloroplasts, which I cannot find. Okay, so it's here. Chloroplasts, which animal cells don't have. Animal cells have, okay, it's not labeled here, but let me draw it myself. Very close to the nucleus. Yeah, it's here. There will be structures called centrioles. They are present only in animal cells. You can not find them in plant cells. Otherwise, the plasma membrane, mitochondria, nucleus, ribosomes, all of these are similar, but it's not identical, means it's not exactly the same. Some differences will be there. Cell wall is present only in plant cells, chloroplast or plastids in general only in plant cells, and large central vacuole only in plant cells. And in animal cells, you have centrioles, which plants don't have. Okay, so these are some differences. I mean, you know this from your lower grades, right? As always, we'll start from outside to inside. We'll start off with the cell wall. Cell wall is a non-living structure. It is very rigid in nature and it will form the outermost covering in some types of cells like plant cells and fungal cells. Okay. What is its function? The function of a wall anywhere is the same. I've told you this. It provides the shape. It protects the cell. Here we are talking about multicellular eukaryotic organisms also. So if the organism is multicellular, neighboring cells will have to interact with each other while forming tissues. So since this is the outermost wall, it allows for cell to cell interaction and it also provides a barrier for undesirable molecules. It will not allow undesirable molecules to enter the cell. Now, I told you that plants have cell wall, fungus has cell wall, right? What is the cell wall made up of? Is it the same in all these eukaryotic organisms? In algae, generally, it's made up of cellulose, of course. Algae or any plant, cellulose will be present. In algae, you can find galactans, mannans, and minerals like calcium carbonate. But in higher plants like bryophytes, pteridophytes, angiosperms, gymnosperms, you will find cellulose, hemicellulose, pectin, and different types of proteins. In fungi, it is a characteristic feature that the cell wall is composed of chitin. We've learnt about it in biological classification, right? I hope you remember. So what is the structure of a plant cell wall? That's what you have to study. In higher plants, what is the cell wall like? In higher plants, the cell wall can be of many different layers. Let's take a plant cell for example. Okay. So this plant has a cell wall. This plant has its nucleus and genetic material. Let's say this cell decided to undergo cell division. It has made an extra copy of its nucleus. Now this cell cytoplasm has to divide. When the cell cytoplasm has to divide, something called a cell plate will form. Okay, it starts forming in the center and it will continue towards the periphery. Okay, so this cell plate is the first layer of the cell wall that forms. This is called middle lamella. This is called middle lamella. Now, in plant cells, only some cells will retain the ability to undergo cell division. We call them meristematic cells or meristematic tissues. So, eventually, some of these cells will stop dividing and become mature. They will start to get differentiated. We've learnt about it in anatomy of flowering plants, right? We've learnt that some cells, they modify according to which type of cell it's going to become in the future after it gets differentiated. Like sclerenchyma cells, they lose their protoplast and they develop a thick lignified wall. We've learnt about it. First cell division happens, then cell elongation happens, then cell maturation happens. So right now cell division has happened. After cell division has happened, firstly a cell plate was laid during cytoplasmic division. That is called the middle lamella and that is composed of calcium pectate. Okay, right now, if this cell has to synthesize additional wall layers, if more wall layers have to be added, then that substance has to be synthesized at the cytoplasm itself. So, it will be synthesized in the cytoplasm itself. So, outside the middle lamella, nothing can form. Towards the cytoplasm only more layers can form because whatever substance is required for cell wall formation is synthesized right here. So, it's only towards the inside that newer wall layers can form. Alright, so let's take this one cell right now. It has just formed. It is a baby cell. Right now, all it has is the middle lamella made up of calcium pectate. Now, this cell is growing in size. As it grows in size, it will start synthesizing a layer of primary cell wall inside. Right inside this, it will start synthesizing a primary cell wall. This is the middle lamella. Inside the middle lamella, you will have the primary cell wall that is synthesized. Okay. This is the primary cell wall. This is generally made up of cellulose, hemicellulose and pectin. Okay. This is present in all types of cells, parenchyma, collenchyma, everywhere this cell wall layer will be present. In addition to this, only in some types of cells, you can find the presence of another layer called the secondary wall. The secondary wall is present only in some types of cells. It's not present in all types of cells. This may be made up of substances like lignin, as in the case of sclerenchyma. Are you getting my point? When every type of cell undergoes, cell undergoes cell division, that is meristematic tissues undergo cell division and newer cells are formed, during cytoplasmic division, first nucleus will divide, and when cytoplasm is dividing, first a cell plate is laid. That cell plate itself is the middle lamella. That is the first layer that connects to neighboring cells. Okay, that separates two neighboring cells, I mean. That is called the middle lamella. It's made up of calcium pectate. It is what cements two neighboring cells. Now these meristematic cells that have newly formed, if they stop undergoing cell division, they will want to differentiate and become permanent tissues like parenchyma, collenchyma, xylem, phloem, whatever. If they want to get differentiated, before differentiating, first they will increase in size. As they increase in size, they will form a primary cell wall made up of cellulose, hemicellulose, pectin. As the length of the cell or the size of the cell increases, newer cell wall material, hemicellulose, cellulose and all of that will get accumulated and the cell wall will increase. Okay. In different types of plant cells, you will have different thickness of the primary wall. In some the primary wall is thin, in some it is very thick. In meristematic tissue, we've learned that they have very thin cellulosic cell walls. But in some differentiated cells, the cellulosic cell wall will be thick. Why I am saying cellulosic is because cellulose is the primary component there. Of course, hemicellulose and pectin will be there, but cellulose parallelly will be arranged and that is the major component in these primary walls. Depending upon which type of tissue or which type of cell this is going to get differentiated into, it may form an additional secondary layer towards the cytoplasm. And for example, in the case of sclerenchyma, it is composed of a substance called lignin. So inside this, you will have the plasma membrane. Inside that, you will have the plasma membrane. Inside the plasma membrane, you will have the cytoplasm. Okay. Now, between two neighboring cells, if they have to form tissues, they will have to communicate with each other. So if this is one cell and this is another neighboring cell right here, it will have its middle lamella, it will have its own primary wall, it will have its own secondary wall, it may have its own secondary wall also, and it will have its plasma membrane as well. I'm showing it only on one side, but know that it will be present throughout. For ease, I'm showing it only on one side. Now, in between this cell and this cell, if communication has to happen, communication happens by means of chemicals. For those chemicals to talk to each other, move from this cell to this cell, there has to be some provision that is provided by cytoplasmic connections called plasmodesmata. So, in between these two cells, in certain regions, you can see the presence of cytoplasmic connections called plasmodesmata. They connect the cytoplasm of one cell with the other cell. So through that, because of the interconnected cytoplasm, communication can happen between these two neighboring cells. I hope you've understood. This cytoplasmic connections here that exist between these two cells, this is called plasmodesmata. Okay, I hope it is clear. So when you're asked from outside to inside, what are the different layers? Outermost will be the middle lamella. First, outside to inside, out to in. First will be middle lamella. Inside the middle lamella, you will have primary wall. Inside that, you will have secondary wall. This is only in some cell types. Inside that, you will have plasma membrane. Just because middle lamella is called middle lamella, you shouldn't think that is present in the center. Between primary and secondary, it's present in the middle. You should not think like that. It's called middle lamella because when it formed, it was in the middle of two different cells that divided. That's why it's called the middle lamella. Okay, so that's about the cell wall in plants. Now let's talk about the cell membrane structure. This structure, though we are learning under eukaryotes, this structure is very similar even in prokaryotes as well. And here we are learning this as a structure of the cell membrane, but this structure is true for the membranes of organelles as well. When we say lysosomes are bound by membranes, nucleus has a membrane, mitochondria has a membrane, ER has a membrane, the structures of those membranes are also very similar to the structure of plasma membrane. So right now we are studying about the structure of biological membranes. Alright, now what is our cell membrane composed of? Our cell membrane is primarily composed of lipids. It is composed of a type of lipid called phospholipid mostly. Alright. Now how are the phospholipids present? The phospholipids will have two parts. Each phospholipid molecule will have two parts. It will have a head containing the phosphate group. You call that the phosphate head. To this phosphate head, two hydrocarbon tails will be attached. Okay, so this is one phosphate head and these two here these are called the hydrocarbon tails. Basically, they are the hydrocarbon tails attached to the phosphate group. Okay. Now, these phosphate heads are hydrophilic, which means they like water. But the hydrocarbon tails, they are lipids, no? Lipids don't like water. This part of the lipid does not like water. So these are hydro means water, phobia means fear, philia means liking. Hydrophobic, they do not like water. Okay. Majorly in the plasma membrane, you will find phospholipids only. So in this phospholipid, you have two parts, one part which likes water, one part that does not like water. Now, if this is a cell, inside the cell, you will have the cytoplasm, which contains a lot of water. Outside the cell, you will have intercellular or interstitial fluid, which also contains a lot of water. Now, within this membrane, this molecule is present in large quantities. Basically, the membrane majorly is made up of this type of lipid only. Now, if it's present with the hydrophilic head on this side and hydrophobic tail towards the inside, then inside also there is water. This hydrophobic hydrocarbon tails, they are non-polar, they do not like water. So this will not work. If it is arranged like this, then also it will not work because outside also there is water. So basically within this membrane, you can see that the phospholipids are arranged in two such layers, such that within the plasma membrane, outside also the hydrophilic heads are facing, towards the inside also hydrophilic heads are facing. In between, the hydrocarbon tails which are hydrophobic are present. So basically the phospholipid exists as a bilayer over here. Okay, because there are two layers, it's called a lipid, a phospholipid bilayer. How did we get to know that membranes are made up of phospholipids? Earlier investigations were biochemical. Electron microscopy was not very advanced. So microscopy of course was not very advanced. So they could not look at the structures of cell membrane under the electron microscope, but they made biochemical analysis and found out that the plasma membrane is mostly made up of phospholipids, and then it is arranged in the form of two layers. Okay. Phospholipids are the major type of lipids you find in the plasma membrane. That's not the only type of lipid. Along with phospholipids, you also have lipids like cholesterol, which provides a lot of stability to your plasma membrane. Okay, so this is the structure of the plasma membrane given in NCERT. You can see how all of these blue colored molecules, which now I will outline in pink color, it has head on the outside and the tails that are hydrophobic are facing the inside. So there is a bilayer of lipids. Further investigations, biochemical investigations, we got to know that within the plasma membrane, proteins are also present. So lipids are one component. Along with lipids, proteins also are present. So these proteins are basically present within the phospholipid bilayer. Depending upon how easily you can extract that protein from the membrane, you can classify it into two types. You have integral proteins and peripheral proteins. I'll show you what the difference is. I want you to notice this one protein right now. This protein is present spanning through the entire plasma membrane. It's completely integrated or present within the plasma membrane. Such proteins are called integral proteins. They are difficult to extract from the membrane. There are some other proteins like this one here. This is also partially embedded. Okay. If they are partially or fully embedded within the membrane, you call them integral proteins. Some proteins lie peripherally like here. Okay, on the surface. I'll show you. Yeah. So some of them may lie on the surface superficially like this. They are called peripheral proteins. They are relatively easier to extract. Okay, is that clear? So depending upon whether it can be extracted from the plasma membrane phospholipid bilayer easily or not, we have two types. One is integral, the other one is peripheral. Even if it's partially buried inside like this or completely present within the phospholipid bilayer, it is integral. If it is present superficially, then you call it peripheral protein. Okay? Yeah. Now, depending upon which cell type it is, the proportion of lipids and proteins in a membrane can vary. If you consider our human RBCs, for example, or erythrocytes you can call them, in our human RBCs, which is majorly used for the study and analysis of cell membrane structure, the protein content is more than the phospholipid content. So the protein is about 52% while the lipid is just 40%. Okay. So proteins are 52%. Lipids are about 40%. That varies from cell to cell. Apart from this, small amounts of carbohydrates also are present in the membrane. Okay. These carbohydrates, as you can see here, these chains, they may be associated with the lipids or they may be associated with the proteins. If the carbohydrate is associated with the lipid, then the lipid is called glycolipid. If it is associated with the protein, then the protein is called a glycoprotein. So in a plasma membrane, you find lipids, majorly phospholipids, some cholesterol, proteins of two different types, and also some carbohydrates associated with lipids or proteins. Okay? Yeah. Now let's talk, whatever I've explained so far, I have written down in text here. I've typed it all for your studies later. You can use it. It'll be available in your notes. Yeah. Now, two people, Singer and Nicholson, they proposed a model for the plasma membrane called the fluid mosaic model. It was proposed by them in the year 1972. What they basically say is the plasma membrane is not rigid. It is a quasi-fluid. It is quasi-fluid in nature, means semi-fluid. It has oil-like consistency, lipid-like consistency, and within that, whatever proteins are there, Because of this fluid-like nature of plasma membrane, it is possible for these proteins to move sideways. Imagine if the plasma membrane phospholipids were solid, if they were solid structures, very rigid structures, the proteins would not be able to move within them. Now, we say that the plasma membrane is kind of fluid, quasi-fluid, semi-fluid, which allows the movement of these proteins sideways within the membrane. That is called the fluid mosaic model. Basically, they say that the plasma membrane is quasi-fluid in nature. It is quasi-fluid because of the presence of the lipids basically. Okay? Yeah. Because these proteins can move within the membrane, we say it is fluid. Why is it mosaic? Because in a common background you have common background of phospholipids, you have patch-like proteins present. That's why we say it is mosaic. Okay? Yeah. What are some functions of the cell membrane? Transport of many important molecules happen across the cell membrane into and out of the cell. This transport can be categorized into two types broadly. One is passive transport, the other one is active transport. Passive transport happens along the gradient. The molecules are transported from where it is more to where it is less. It doesn't require ATP. This can happen in three types. One is simple diffusion, second is osmosis, third is facilitated diffusion. Osmosis is diffusion of specifically water. Okay, so let's see what these three types are first. Simple diffusion, if there is more molecules of one type on one side and the other side there is less, because of the concentration gradient, if these molecules are not polar, if they are non-polar, then directly by dissolving through the phospholipid bilayer, by coming through the phospholipid layer, they can reach the other side of the membrane. If they are polar molecules, then they cannot come directly via the phospholipid bilayer. There will be special proteins for them present in the membrane. These membranes will help in the transport of this substance from a region where it is high to a region where its concentration is low. Alright, so that is called facilitated diffusion. Diffusion is happening, passive transport is happening, but that is facilitated by this protein. So we call it facilitated diffusion. If there is diffusion specifically of water molecules across the membrane, generally it happens through aquaporins present in the membrane, then we call it as osmosis. What is common to all these three is that movement happens from a region where there is more to a region where there is less concentration. In active transport, it's the opposite. Here it happens against the gradient. Movement happens from a region where there is already more of that substance to a region where its concentration is less, and this 100% requires ATP because it has to be pumped, it has to be against the gradient, right? One example for that given in your textbook is your sodium potassium pump. This is a type of protein present in the surface of your, within the plasma membranes, as you can see here. Here, against the concentration gradient, ATP is utilized and molecules are transported. Three ions, sodium ions are transported against the gradient outside the cell. In exchange, 2 potassium ions are pumped into the cell by making use of 1 ATP. In both cases, it is happening against the concentration gradient. From wherever it is already more, it is getting pumped. From a place where it is more is going. Okay? From a place where it is less, more is going. I think I wrote it ulta here. Let me just change it off. Yeah. This is wrong. It's going from a place where it is less to a place where it is already more. Less to more. Okay, that's why it requires energy. It consumes energy in the form of ATP. Yeah, the functions I've written about all the different types I've explained so far. Now let's talk about the endomembrane system. What is the endomembrane system? You know that in eukaryotic cells, we have many different types of organelles present inside the cell, such as mitochondria, chloroplast, endoplasmic reticulum, Golgi, lysosomes and all of that. Now, some of the membrane-bound organelles present inside the cell, inside, membranous structures, membrane-bound organelles. Some of these, not all of them, not all the membrane-bound organelles, some of these organelles and their functions are coordinated. All of those organs whose functions are coordinated within the cell, together we call it the endomembrane system. It includes 4 different types of organelles. Firstly, the endoplasmic reticulum, then Golgi apparatus, lysosomes and vacuoles. Okay? We'll have to learn about each of these first and then we'll learn about the other organelles. First, we learn individually about how is their structure, what is their function, and then I'll tell you how their functions are coordinated. Okay? We'll start with endoplasmic reticulum. Endoplasmic reticulum, endoplasmic means what? Inside the cytoplasm. Reticulum means what? Network. So within the cytoplasm, endoplasmic reticulum forms a network-like structure. That is why we call it the endoplasmic reticulum. Okay, so it is network or reticulum. All right. The endoplasmic reticulum is nothing but tubular structures that are scattered within the cytoplasm. Generally, it arises from the nucleus. As you can see here, these are extensions that arise from the nucleus like this. And sometimes it goes up until the plasma membrane as well. Basically, this is present in the cytoplasm. If you think of this cell, this as a cell, this is the cell membrane. And inside the cell membrane, you have the cytoplasm. The presence of this endoplasmic reticulum divides the cytoplasm into 2 compartments. As you can see, inside the endoplasmic reticulum, there is space, right? Here, all this is space. Let me use a different color. See, inside this orange color, there is space here. That space that is present inside the endoplasmic reticulum is what we call the lumen of the ER. This space is the lumen of ER. The presence of these membranes in the endoplasmic reticulum divides the whole cytoplasm into 2 compartments. One is this lumen that is there, which we call the luminal compartment, which is inside the membrane of the ER. And outside what you have, that is called the extraluminal compartment. Like if you think of this membrane, towards the inside of the membrane you have lumen, towards the outside of this membrane is called the extraluminal compartment of the cytoplasm. Right? Now this ER is of 2 types, smooth endoplasmic reticulum and rough endoplasmic reticulum. These 2 types are based on whether on their surface ribosomes are present or not. If ribosomes are studded on top of the on top of the ER, it gives it a rough appearance. So we call it the rough endoplasmic reticulum. If it does not have any ribosomes present on it, then we call it the smooth endoplasmic reticulum. Okay, so the rough endoplasmic reticulum is associated with ribosomes. Smooth endoplasmic reticulum is not associated with ribosomes. The function of the rough endoplasmic reticulum is to help in protein synthesis. They themselves do not synthesize proteins, but since there are so many protein factories or ribosomes sitting on top of it, like these ones, all of these are ribosomes, their job is to synthesize proteins. It's creating a surface area for all of these ribosomes to synthesize their proteins. So, these rough endoplasmic reticulum, it will be found in mostly cells where protein synthesis is actively happening. Smooth endoplasmic reticulum is the site where lipid synthesis happens. Okay, and these are found mostly in cells where steroidal hormones and stuff are synthesized, because steroidal hormones are also lipids. So you will find more of these smooth endoplasmic reticulum present in such cells. This will continue from the rough endoplasmic reticulum. Rough endoplasmic reticulum will continue from the outer membrane of the nucleus. This is the difference between these two types. That's all you need to know about ribosome. Rough endoplasmic reticulum generally. Okay. Now the next organelle that we have to learn about is the Golgi apparatus. It's called Golgi apparatus because it was first discovered by a scientist called Camillo Golgi in the year 1898. He saw that the Golgi apparatus usually is present very close to the endoplasmic reticulum. It is made up of very flattened sac-like structures that are arranged parallel to each other. He calls these flat sac-like structures as cisternae. Where is it? Yeah. They generally measure about 0.5 to 1 micron in diameter. As you can see here, this is one sac, flat sac. It also has lumen inside, right? So you have many such sacs that are arranged parallel to each other like this. Okay. So these flattened sac-like structures are called cisternae. Usually these are present within the nucleus, present near the nucleus and endoplasmic reticulum only. This has a very clear convex side and a concave side. If you think of this as the cell, here is the nucleus, here the ER is there. Okay, near the ER usually this will be present. So it will have a concave side. This is the convex side, and it will have a clear concave side. If it's like this, one side will be convex. This side is the convex side and this side is the concave side, right? So it has two distinct convex and concave sides. The convex side always is towards the nucleus and endoplasmic reticulum. This convex side is also called the cis phase or forming phase. This concave side is called the trans phase or maturing phase. That is its structure. Now let's talk about its functions. What is the function of Golgi apparatus? It is involved in packaging material that have to be transported somewhere within the cell or somewhere out of the cell. Okay. I'll tell you how endoplasmic reticulum's function is related to this organelle's function with the help of this diagram. I'll show you. Here you see, this is the endoplasmic reticulum, rough endoplasmic reticulum. On the rough endoplasmic reticulum, you have ribosomes. Ribosomes synthesize proteins. Let's say the ribosome has synthesized some protein to which sugar has to be added. Remember, I told you there is in plasma membrane certain proteins which have carbohydrates attached to it. So to that protein which the ribosome has synthesized, carbohydrate has to be attached. So this ribosome sitting on top of the ER, let's say this is the ER, inside this there is space, that is lumen. Sitting on top of this, the ribosome will synthesize the proteins. The proteins are transported into the lumen of the mitochondria. As you can see here, let's say these are the proteins that were synthesized by the ribosome. So this ER pinches this off in the form of a vesicle. The membrane gets enfolded there like that. The membrane gets pinched off, a small ball or vesicle forms carrying the protein to which the carbohydrate has to be added. This will come here and fuse with the cis phase of the Golgi membrane, Golgi apparatus, which is the convex side. Forming phase will come and fuse. As it moves down the Golgi apparatus, Golgi will add sugars to it by a process called glycosylation. Once it reaches the trans phase, to the protein, sugar may have gotten added already. Now the Golgi will pinch it off in the form of a vesicle and it gets transported to the plasma membrane or wherever it is required in the cell. Okay, so the function of this and this is coordinated. All right. Yeah. So it's involved in packaging, and whatever materials have to be packaged in the form of vesicles, it will come to the ER in the form of vesicles and fuse with the cis phase of Golgi apparatus and move towards the maturing phase. Many proteins synthesized by ribosomes on the ER are modified in the cisternae of Golgi apparatus before they are released from its trans phase. So, it is important site for formation of glycoproteins and glycolipids. If glycoproteins have to form, to the protein, sugar or carbohydrate has to be added. If glycolipids have to form, sugar or carbohydrates have to be added to the lipid. So, if it has to form a glycoprotein, it will receive a vesicle from the ER. If it has to form a glycolipid, it will receive a vesicle from the smooth ER, because that is where lipid synthesis happens, right? So it will modify that protein or lipid within its cisternae, and once it is modified, it will release it in the form of a vesicle within a vesicle from the trans phase. Okay? Now, let's talk about the next organelle in the endoplasmic reticulum, in the endomembrane system, which is the lysosome. Everybody knows lysosome as the suicide bag of the cell. Lysosomes are called so because they contain a lot of hydrolytic enzymes that have the ability to break down pretty much any biomolecule. So it has enzymes like lipases, proteases, carbohydrases, nucleases and all of that. Lipases are enzymes that break down lipids. Proteases break down proteins, carbohydrates breaks down carbohydrates, nucleases break down nucleic acids like RNA and DNA. So many different types of these enzymes are present within the lysosome. That's why they're called suicide bags, because this can break down all of those molecules. These enzymes are activated only at an acidic pH of about 2, all right? Yeah. How these lysosomes form is interesting. Lysosomes have a lot of enzymes present within that. All of these enzymes are proteins. They have to be synthesized in the ribosome. They are synthesized by the ribosomes present on ER. ER will form a vesicle containing those enzymes and send it to the Golgi. Golgi will modify them and release a vesicle containing all of these enzymes. That itself is the lysosome. So if they ask you for lysosomes are formed from which organelle, then you should know they're talking about Golgi apparatus, because lysosomes get pinched off from the Golgi apparatus itself. It is called suicide bag because its function is to destroy things that are not working well in the cell. It could be a damaged organelle. For example, mitochondria, if they become old, they're not producing as many ATP as they should. The plasma membrane, the membrane of the lysosome fuses with the outer membrane of the mitochondria. Normally, pH will not be 2. So these enzymes won't be active. They will be in their inactive form. Once this fusion happens between a damaged organelle and the lysosome, protons will be pumped inside so that the pH will come down. It will become acidic. And that is when these enzymes will get activated. They will break it down. Okay? Yeah. Also, if amoeba or something engulfs a food vacuole, for digestion, the food vesicle also combines with the lysosome and helps in the digestion. So that's what the function of lysosome is. Now let's talk about the last part of the endomembrane system, which is the vacuole. You mostly find it in plants, right? In plant cells, you have a cell wall, you have the cell membrane, and within the cell membrane, you have the cytoplasm, and you have a large central vacuole in plants. The nucleus also goes off to one side. This is the vacuole. The membrane of the vacuole is called the tonoplast. Within the vacuole, you have sap, which contains a lot of ions and waste material. Generally, the concentration of ions in the sap is much higher because from the cytoplasm also, ions will be pumped inside, making use of energy. That is the reason why even within the vacuole, the sap ion concentration is high. In plant cells, the vacuole can occupy as much as 90% of the volume in the cell. Okay. So vacuole is bound by a single membrane. It's called the tonoplast. It contains water, sap, excretory products and other material that's not useful for the cell, waste material basically. It can occupy as much as 90% of the volume of the cell. Okay. It facilitates the transport of a number of ions and other materials against their gradients into the vacuole. Therefore, the concentration is significantly higher in the vacuole compared to the cytoplasm. In amoeba, you have a type of vacuole called contractile vacuole, which helps in osmoregulation and excretion. Basically, amoeba are aquatic organisms. They grow in usually fresh water. So, if they are surviving in fresh water, remember amoeba is an animal cell, they don't have a cell wall. Because there is a difference in water potential, the cytoplasm is more concentrated compared to the surrounding water. By osmosis, by endosmosis, water can enter the amoeba. There is no protective rigid cell wall. So this amoeba, if there's a lot of water entering it, it can rupture and break. To prevent that, what happens? Excess water is stored in a contractile vacuole and it is expelled out of the amoeba all the time. That's called a contractile vacuole. If the amoeba engulfs a food particle, it forms a vesicle which is called the food vacuole. Okay, so these are some examples. Now let's learn about other organelles which are not part of the endomembrane system, starting with mitochondria. Mitochondria is called the powerhouse of the cell, something that you will never forget. The reason is, it is the organelle in which majorly ATP synthesis takes place. Okay, mitochondria is plural, mitochondrion is singular. This is not very easily visible under your compound microscope. The number of mitochondria in a cell varies. If a cell is metabolically very active, then more mitochondria will be present compared to cells that are not very active physiologically. Its shape can vary from a sausage shape to cylindrical. Its size in diameter is about 0.2 to 1 micron. Length can vary between 1 to 4.1 microns. Let's see its structure. It's bound by two membranes. It's bound by two membranes. Okay. You have a smooth outer membrane and an inner membrane which has a lot of infoldings like that. Let's label it. This is the outer membrane, which is smooth. Inner membrane has infoldings that are called cristae. Within the inner membrane, you have the matrix. Within the matrix, you may have small circular DNA. Within the matrix, you can also find ribosomes. But these ribosomes, unlike eukaryotic ribosomes, are of 70S type, similar to bacteria. Okay? The reason why there is so many infoldings in the inner membrane is because in the inner membrane, surface area is increased. I've already told you when I explained mesosomes. There are enzymes involved in the synthesis of ATP present here. We say that mitochondria is the powerhouse of the cell. It produces a lot of ATP molecules and all of that, right? So for ATP molecule production, these enzymes are required. If the enzymes are more in number, more ATP will get produced. If you want to accommodate more such molecules, ATP synthase enzymes to synthesize ATP, surface area has to be increased. In order to increase surface area, in the inner membrane, you can see a lot of infoldings. The space present between the outer membrane and the inner membrane is called intermembrane space or perimitochondrial space. Okay, so that's the structure of mitochondria. Mitochondria has its own ribosome, it has its own DNA. It will not depend on the genomic DNA to replicate. It can undergo division like binary fission in bacteria. Okay, so we say that mitochondria is a semi-autonomous organelle. For the synthesis of ATP, some of the enzymes it can synthesize by itself by the ribosomes that are present here. The information for that will be present in the DNA here. It does not have to depend on the genomic DNA for it. So we call it a semi-autonomous organelle. Next, plastids. These are found in photosynthetic eukaryotes such as plant cells and euglenoids. Compared to mitochondria, these are slightly larger and they can be seen better under the microscope. These have specific pigments, and because of those pigments, different plant parts appear different colors, like flowers have one color, fruit has one color, leaf has one color. That's because of the type of pigment present in these plastids. You have three different types of plastids based on which pigment is present. Most popular one is chloroplast, then we have chromoplast, and then we have leucoplast. Chromoplast contains the green pigment chlorophyll. It will contain some carotenoids as well, but majorly green color is because of chlorophyll. Chromoplast imparts color to different plant parts like flower petals and fruits. So they contain pigments other than chlorophyll, such as carotenes and xanthophylls, yellow, orange, red colors. These pigments are fat soluble, they are not soluble in water. Leucoplasts, leuco means colorless. So these leucoplasts are colorless, they are majorly involved in storage of nutrients. Depending upon which type of storage food is there, you have three different types of leucoplast: amyloplast, aleuroplast, elioplast. Amyloplast stores starch like carbohydrates. Carbohydrates like starch. Elioplast stores oils and fats, basically lipids. Aleuroplast stores proteins. We have to learn the structure of chloroplast only. So, chloroplast is mostly found in the green parts of the plant. In the mesophyll cells of the leaves, we know about it. Its shape varies from lens shape to oval to spherical to discoid and sometimes even ribbon-like. We've learnt about this in the chapter Plant Kingdom as well under algae. Size is variable, slightly larger than mitochondria, I told you. The length varies between 5 to 10 microns and the width varies between 4 microns. Number is variable. In some cells, per cell there will be only one chloroplast, as in the case of Chlamydomonas. But in the mesophyll cells, you can find as many as 20 to 40 chloroplasts. Let's understand the structure of chloroplast. Similar to mitochondria, this also is bound by two membranes. This also is a semi-autonomous organelle. Okay. It has two membranes. Both the membranes are smooth here. There is no infolding or anything. Okay. This is like a section. This is your outer membrane. And inside you have the inner membrane. The space between these two is called periplastid space or intermembrane space. The matrix inside is granular and it's called the stroma. Within the stroma, you have additional membranous structures called thylakoids. Each such flat membranous sac is called a thylakoid. You will have many thylakoids stacked one on top of another to form a stack called the granum. Granum is singular, grana is plural. Within the matrix, you can find many such grana. grana are interconnected with each other through membranous structures again. These are called stroma lamellae. Within the stroma, you can find circular DNA and also ribosomes, similar to how we saw in mitochondria. And these ribosomes are similar to prokaryotic ribosomes, which means they are 70S type. Why are these extra membranes present? I told you that even in the chloroplast, pigments are present within the membranes. They are lipid soluble, right? They are soluble within the phospholipid present in the membrane. So in order to increase the surface area to accommodate all of those pigments, these extra membranous structures are present. So within these grana and stroma lamellae, you can find pigments present. We learn more about it when we discuss the chapter photosynthesis in higher plants. Okay, for now, for this chapter, this is all you need to know. Now, let's talk about ribosomes in eukaryotes. What we already know is that they are not bound by any membrane. It's common to both prokaryotes and eukaryotes. They are also called pallid particles because it was first discovered by a person by the name George Pallid in the year 1953. So if it's not bound by a membrane, what is it composed of? It is composed of a type of RNA called ribosomal RNA. In short, we call it rRNA. Along with that, you will have many proteins. That's all the ribosomes are composed of. So you will have some types of RNA. Along with that, there will be some proteins associated. This is one subunit. Like that, there will be another subunit. So you have the two subunits of RNA made ribosome made up of just ribosomal RNA, a type of RNA, and proteins. Talking about eukaryotic ribosomes, in eukaryotes you have ATS type of RNA, ATS type of ribosome. The ribosome that you find on the surface of ER is ATS. What you find in the cytoplasm is ATS. What you find on the surface of nucleus is ATS. But what you find in chloroplast, in mitochondria, that is 70S. In ATS ribosome, the large subunit is 60S and the small subunit is 40S. Together it is ATS. Okay, S is Swedberg unit or sedimentation coefficient. I've already told you about that. So the large subunit is 70, small subunit is 40. Remember, all eukaryotic ribosomes are not ATS. The ones that are present within the chloroplast and mitochondria, like I've already told you earlier, are of the prokaryotic type, which means it is 70S. Cytoskeleton. What is the cytoskeleton? Similar to how our body has a skeletal structure made up of bones and cartilage, every one of our cells also has a skeleton of its own, which is called the cytoskeleton. Cyto means cell. So unlike our skeleton, which is made up of bones and cartilage, the cytoskeleton in cells is made up of proteins. There are three different types of protein that make up the cytoskeleton. They are the microtubules, microfilaments, and intermediate filaments. Okay? They appear like this inside a cell. This here is the cell membrane. Within that you can see these green color tube-like structures. They are your microtubules. These orange network-like structures, they are the intermediate filaments. And these red color structures present towards, very close to the plasma membrane, they are your actin filaments. Or you can say microfilament. Microtubule, actin filament or microfilament, and intermediate filament. Okay? Yeah. So what is the function of this cytoskeleton? Similar to the function of our body skeleton, it helps in locomotion, it helps in movement, right? It helps in providing a specific structure to the body. This provides specific shape and structure to the cells. It gives mechanical support. This also provides mechanical support. So the same three functions that our skeleton does. Mechanical support helps in motility, movement and stuff. And maintenance of the shape of the cell. We saw how shapes can be so different, right? Cuboidal, squamous and all of that. So how is that possible? That is maintained by these cytoskeletal elements only. Okay, so that's about your cytoskeleton. Next we have locomotory structure, cilia and flagella. In prokaryotes also we learnt about the structure of flagella. That was very different from how the structure of flagella is in eukaryotes. Both of their function is the same, they are analogous. So both of them help in motility, movement, but they are structurally very different. In eukaryotes, cilia and flagella, both of them are covered by a plasma membrane. In organisms like paramecium, you can see many cilia arising from the surface. In organisms like euglena, you can see flagella present in eukaryotes. Both of them help in the process of locomotion. They are hair-like outgrowths that arise from the cell membrane. Cilia are many in number but shorter. Flagella are very few in number but relatively longer. These flagella beat together, similar to how you see boat race in Kerala. All of the people sitting are rowing the boat at the same time. They are rowing their oars at the same time. Similarly, these will beat in the same direction and it will propel the organism forward. So, yeah. I already mentioned how the cilia and flagella both are covered by the plasma membrane. They are enclosed by the plasma membrane. Here's what its structure looks like. This is one flagellum, let's say. Cilia also structure is the same. Outside what you're looking at, blue color, that is the plasma membrane covering. The core that is present inside, made up of microtubules, made up of proteins, that is called the core or the axoneme of the flagellum or cilia. Okay, it's called the axoneme. It's made up of microtubules. Where else have you heard about microtubules? I just told you they are one of the cytoskeletal elements present in the cell, right? So it's the same microtubules. Its arrangement is unique. It is arranged in the 9+2 array, which means I want you to understand that each microtubule, if this is a flagellum, each of these circles that you're looking at here are microtubules that are present here. You're looking at it in the form of circles because you've taken a section of the flagellum and you're looking at it under the microscope. Inside the flagellum, outer boundary or outer covering, you have the plasma membrane. In the core, you have many tube-like cylindrical structures present, microtubules. When you cut it and look at it, it will appear circular, right? So these are circles, but in the cilia or flagella, they are cylindrical structures that run throughout the length. You can see there are 9 doublets and 2 singlets. 9 doublets are peripherally arranged. Doublet means 2 microtubules present together. Over here you can see this and this. There are 2 microtubules, right? So 2 are present here. In the peripheral region, you can see 9 such doublets. This is called a doublet. So you have 1 and 2, 3, 4, 5, 6, 7, 8, 9. You can see 9 such doublets. In the center, you can see 2 singlets. That's why you call it 9 peripheral plus 2 central microtubules. Okay, these are the peripheral doublets, peripherally located doublets and centrally located singlets. There's 2 of them. Peripherally located, 9 doublets are there. Okay. These 2 are the central singlets. Surrounding that, you have a sheath that is called the central sheath. To this, there are radial spokes that connect the peripheral doublets, as you can see here, similar to how you have spokes for a wheel. There are spokes that are radial and connect the peripheral doublets to the central sheath in the center. The 2 singlets in the center are also connected by what is called a bridge. And the peripheral doublets are also connected to each other by means of what we call the linkers. This is the structure of microtubule. This is the structure of axoneme of flagella and cilia. There are made up of microtubules. There are 9 doublets peripherally, 2 singlets centrally. The 2 singlets are connected by a bridge and are covered by a sheath. The peripheral doublets are connected with the neighboring ones with each other with the help of linkers. The peripheral doublets are connected to the central sheath by means of radial spoke. This is called the 9+2 array of arrangement of microtubules in flagella and cilia in eukaryotes. In prokaryotic flagella, you just had hook, basal body and filament. Here, it is entirely different. It's more complicated. Okay. Yeah. So, I'll label these linker, 9 radial spokes, bridges, central sheath, all of that. Okay. So, these are the important keywords you must remember. Let's talk about centrosome and centriole. Centrosome is a structure that you find only in animal cells. In each centrosome, you can find 2 perpendicularly arranged centrioles like this. See, this is one, this is one. They are almost always present near the nucleus only. It contains 2 cylindrical structures. Each one is a centriole. Together, both of them is called the centrosome. Apart from the centrioles, the centrosome also has certain amorphous material surrounding it, which is called the pericentriolar material. Centrioles lie perpendicular to each other. Its arrangement also is like the cartwheel. The centrosome is also made up of microtubules. This is not bound by any membrane. Okay. So, its arrangement is called 9+0. Okay. It's called 9+0. So, here previously we saw there were 9 doublets peripherally and 2 singlets in the center. Here there is no microtubule in the center. In the center you have something called a proteinaceous hub. Peripherally you have 9 triplets here. That means 3 microtubules present together as one unit. So you call it triplets. Peripherally there are 9 triplets. Centrally you have a proteinaceous structure called the hub. Similar to how you had radial spokes connecting the central sheath to the peripheral doublets in cilia and flagella, over here also you have radial spokes present that connect the central hub to the peripheral triplets. Similar to how there was a linker present between adjacent neighboring doublets, here also you have a linker present between adjacent triplets. Okay, it's the same thing. Radial spokes are made up of protein. Alright, adjacent triplets are also linked. I've told you everything. When these flagella arise from a cell within the plasma membrane towards the inside of the cell, they arise from a structure called, the basal body. This basal body also has a structure similar to the centrosome or the centrioles. Okay, so the basal body of the cilia or flagella, and also during cell division in animal cells, we learn about it in the next chapter. The spindle formation happens. The microtubules will start forming spindle fibers from one region. Over there, organization of spindle fibers, all of that happens from these centrioles only. Okay, so that's the function of the centrosome and centrioles. Now let's move on to the last part of the chapter, which is the nucleus. We already know from the beginning of the chapter that it was first described by Robert Brown in the year 1831. Fleming used a basic dye to stain the nuclear material, and he called it chromatin because it appeared colored. Chroma means color. So when the cell is not dividing, we say that it is an interphase. Okay, more about it in the next chapter we learn. When the cell is not dividing, when it is in interphase, inside the nucleus you can find something called chromatin. Chromatin is nothing but DNA arranged like this. DNA normally is not like this. You know DNA like this, right? So DNA doesn't exist like this inside the nucleus. It will undergo some condensation and exist in the form of thread-like structures, chromatin-like structures. More about this you learn in a chapter called Molecular Basis of Inheritance. For now, just remember DNA doesn't exist like this. It will exist in the form of condensed DNA, which is called chromatin. So when a cell is not dividing, DNA will exist in the form of chromatin. Okay. This chromatin will contain DNA along with protein. DNA is wound around proteins. That's why we say chromatin is nucleoprotein, nucleic acid plus protein. Along with that, inside the nuclear membrane, well-defined nuclear membrane, you have the nuclear matrix. You may have spherical bodies which are not again separated by any membranes called nucleoli. Singular is nucleolus. These are the things that you will find in a nucleus. Let's talk about its structure. The nucleus also is bound by two membranes similar to mitochondria and chloroplast. Okay. You have the outer nuclear membrane and you have inner nuclear membrane. Okay. So this is outer membrane and inside that you have the inner membrane. Okay. These outer and inner membrane may fold upon itself in some regions. Okay. And they form pores that are called nuclear pore. Through this nuclear pore, proteins and RNA can be transported into and out of the nucleus. On the outer membrane, ribosomes may be present. Within the inner membrane, inside the nucleus, you have nucleoplasm. Okay. So this is the fluid matrix that is present within the nuclear membrane. Okay. So this is called the nucleoplasm. Within the nucleoplasm, you can find the presence of DNA in the form of chromatin fiber. It's not like naked DNA. This is called chromatin. Certain regions inside the nucleus take up a lot of stain and appear densely stained. Such regions are called the nucleolus. Basically, these are regions where a lot of rRNA synthesis is happening, ribosomal RNA synthesis. Remember I told you that ribosomes are composed of a type of RNA called ribosomal RNA. That ribosomal RNA synthesis happens here. That's why it's very actively undergoing transcription. That's why it will take up a lot of stain and it will appear darker. Okay. So this is called the nucleolus. These are basically the different parts of the nucleus. In between the outer membrane and inner membrane, whatever space is there, that is called the perinuclear space. Okay, so these are the different parts of the nucleus. There are two membranes that are parallel to each other. It is bound by two membranes. I already told you. There is space between the two nuclear membranes called the perinuclear space, which measures around 10 to 50 nanometer in distance. Outer membrane is continuous with endoplasmic reticulum. I told you it bears ribosomes on it. I have labeled that, I have drawn it also. At many places, nuclear envelope is interrupted by minute pores. I told you how that happens, which is formed by the fusion of those two membranes. Okay. Through these nuclear pores, movement of RNA and proteins will happen. That also I told you. The number of nuclei in a cell is variable. Some cells, most cells have one nucleus per cell, but some cells can have more than one nucleus also. We call that multinucleate condition, and there are a few cells which don't have even one nucleus, such as the erythrocytes in humans, RBCs in humans are enucleated. Sieve tube elements in plants are enucleated. So over there you do not find nuclei. Okay. So that's about the nucleus. When the cell is not dividing, it exists in the form of chromatin. When the cell is not dividing, cell not dividing in interphase. When the cell is in division phase, when it is dividing, cell is dividing, it will form even more condensed structure called the chromosome. This is in the M phase when the cell is dividing. Okay, so chromosome also contains protein around which DNA is wound. It's a super compact structure. In a chromosome, you can find a region where there is a constriction. It is narrow in one region, that is called the primary constriction. Wherever there is constriction, narrowing, that region is called the centromere. Okay, this forms during cell division. On either side of the centromere, you have proteinaceous disc-like structures called kinetochores. Whenever spindle formation happens during mitotic cell division or cell division in general, meiosis or mitosis, the spindle fibers attach to the chromosomes at these kinetochores. So these are the sites of attachment of spindle fibers to the chromosome. It happens at the kinetochore. Now, depending upon how the centromere is, where the centromere is, the position of the centromere in the chromosome, you have 4 different types of chromosomes. If the centromere is in the center, as in this case, centromere doesn't always have to be at the center just because it's called centromere. If it is in the center, you call it a metacentric chromosome. If it's slightly away from the center such that you have a slightly short arm and a slightly longer arm, you call it a submetacentric chromosome. If you have it, the centromere very far from the center, then you have a very short arm and comparatively very long arm of the chromosome. Then you call it acrocentric. The centromere could be present at the tip, at the terminal end also, in which case we call it telocentric. Okay. So these are the different types of chromosomes based on the position of the centromere. Sometimes some chromosomes, in addition to having the primary constriction of the centromere, they will have a secondary constriction. Okay, another narrow region. This is called secondary constriction, and this part is called satellite chromosome. Okay. So this is not there in every chromosome. In some chromosomes, it may be present. That's all about the nucleus. Now let's talk about the last part, microbodies. Microbodies are basically membrane-bound vesicles carrying different enzymes that you can find both in plant cells as well as animal cells. So one example for that is peroxisome, which you find in plant cells. It contains an enzyme called catalase that will help in elimination of hydrogen peroxide. Like it is toxic, right? So it will break it down and convert it into water, like it will do a reaction and convert it into water. So that's the function of catalase enzyme in peroxisome. Similar to peroxisomes, we will have many microbodies, very small membrane-bound vesicles present in plant and animal cells that contain certain enzymes. Okay. Yeah, so that's about it. I hope everything you've understood. And if you have any doubts, you can post it in the comments. I'll answer it as soon as I can. Thank you very much for watching this. Hopefully it has helped. I'll see you again in the next lecture with the next chapter. Take care, keep studying hard. All the very best. Thank you.